I am delighted to have you all in this bright Miami sunny morning back to our seminar series. You will see today that we have launched a new initiative, and this new initiative is to recognize and give the opportunity to early career investigators. So our hope is that you will get to hear from people who have most recently graduated or completed their postdoctoral fellowships or are right in the midst of those, and learn about what they are doing and what excites them, and that you will join them at noon in the Zoom room to ask them all kinds of questions. And those questions don't have to be just about their work, but they can be about, why did you choose to do a doctorate? Why do you have to do a postdoctoral fellowship? What makes you keep going? What do you think maybe the problems that you're going to face that come up as the next steps? How do you make a national networking connection? And so these would be questions to ask them and follow up with. But right now, it is my fantastic pleasure to introduce to you two budding scientists who have already contributed a lot in their early career, and who I believe are on a trajectory of major growth. So the first speaker today for half an hour will be Brittany Gonzalez. Dr. Gonzalez just graduated in December with her PhD right here from our biomedical engineering department. She worked in the CV Putics lab under Dr. Ramaswamy. She got her bachelor's in biomedical engineering from Georgia Tech, and her research is focused on in the tissue engineering world. So she has worked on the creation of a tissue engineered heart valve for children with critical congenital heart valve defects. A typical example of the kind of translational research that is happening in our BME department. The other person who will follow Dr. Gonzalez after she has finished her talk is going to be Dr. Hooi Hooi. I'm not sure I'm going to say the last name right. And Dr. I don't know if I'm showing it right, got her graduated with a first class honors in Bachelor's of Biomedical Sciences from RMIT, which is way across the pond in Melbourne, Australia. And then after she completed that, she came here as a postdoctoral fellow, and she's a joint postdoctoral fellow with Dr. Hutchison's lab in BME and Dr. Agulnik's lab in the medical school. She represents the kind of collaborative effort that we do across the university. And her work, she's going to be talking about how the role of naturally occurring peptide hormones in preclinical animal models. And she recently completed an American Heart Association postdoctoral fellowship. And in her case, she's looking at how certain small molecules can impact vascular calcification, which again, obviously, has to do with this is heart disease. And we are 2 days away from Miami Heart Month. What is Miami Heart Month? It is our own BME feature that we do for the whole month. We're going to talk about cardiovascular aspects and cardiovascular disease, and of course, chocolate the chair. So without much delay, let me introduce to you Dr. Brittany Gonzalez. It's all yours, Brittany.
Okay.
Put your questions in the Q&A, all the members who are listening.
Good morning, guys. Thanks for being here today. And before jumping into some of the research that I've been working on, I thought I would give you guys a little bit of background on how I got to where I am now. And so my interest in engineering started kind of at the end of high school, where we were supposed to pick an elective between anatomy and physiology or engineering. And at the time, I could not even think of the sight of blood. Thinking of dissections would make me nauseous. So I naturally chose engineering as my elective. And that was the best decision I ever made, because that's what really got me into knowing about engineering, about the different types of engineering, and specifically biomedical engineering. And so from there, I went to the Honors College in Miami-Dade for 2 years. And I came across one of my professors for my biology and genetics classes. And he sat me down one day and asked me what my career goals were and what I was interested in. And I told him that I was interested in doing some research, but I didn't really know how or where to get involved. And so he told me he was actually doing some research in collaboration with University of Texas. So I started to work with him. And so this was a little bit more of a computer-based research. So we had a genome of a bacteria that was recently found, and we had to look at the different base pairs and sequences to see what proteins were expressed, as well as what species this bacteria was mostly similar to. So it was a great experience, but it also taught me that I wanted to do more kind of hands-on research, more kind of a wet lab. And so from there, I transferred to Georgia Tech. And here I worked with Dr. Wei Sung in his tissue mechanics lab. And here I was doing uniaxial and biaxial mechanical testing on pericardium tissue. And so we were trying to get the characteristics and the mechanical properties of these tissues to use for TAVR applications or transcatheter aortic valves. And my last semester or my last summer before graduating Georgia Tech, I applied to an REU in Lincoln, Nebraska. So here I worked with Dr. Sanjin Rue, and in his it was a bioflow lab. And here I learned how to make microfluidic plates, as well as these Janus gels or hydrogels of different stiffnesses, so that when you would seed stem cells onto them, they would be able to differentiate due to the different stiffnesses. And so all of these different experiences led me to pursuing my PhD. So I actually graduated from Georgia Tech in December. So I had spring and summer semesters kind of just where I was waiting to hear back from PhD programs. So I decided to come back home to Miami. And instead of working for a couple of months, I decided that I thought I should kind of reach out to labs in Miami and kind of volunteer or see what their labs were about while I was waiting to hear back from PhD programs. And so this is where I contacted FIU, specifically Dr. Ramaswamy, and we decided that I would kind of shadow his lab, volunteer, kind of see what their lab was about. And so I fell in love with the work he was doing and decided to pursue my PhD here. And so just this December, I graduated with my PhD. So now I'm officially Dr. Gonzalez, and I am now a postdoc in Dr. Ramaswamy's lab, kind of continuing some of the work that I did in my PhD, as well as training new graduate students to kind of pick up where we left off. And so I share my story with you guys because I hear over and over again from specifically undergrads that they're not really, like they're very passionate and interested in either research, industry, whatever it is, but they think they're too late to start getting involved. And I'm here to tell you guys it's never too late. And this is one of my favorite quotes where it says, the secret of getting ahead is getting started. So no matter if you think it's too late or you think that you don't have the qualities to do something, just get started. And I promise you that it will be worth it. So now kind of switching gears back to research. Today my talk is on regenerative valves for critical mitral valve disease in the young. So I'll kind of explain some of the work I did in my PhD and then what we're doing continuing forward. So our work focuses on the heart, specifically in heart valves. And as you can see, the heart has 4 valves where the purpose is to promote unidirectional blood flow. And I work specifically on the mitral valve. And here, when the mitral valve is during systole, the valve is going to be closed and the aortic valve is open, which will allow blood to flow to the aorta. And then during diastole, the valve is going to be open. and the mitral valve will be open and the aortic valve is going to be closed. And this is where your heart is filling the left ventricle. And so the mitral valve leaflet has 3 layers and it's highly ordered. So the 3 layers are the fibrosa or the ventricularis, the spongosa, and the atrialis. And you can see here that the fibrosa is mostly composed of collagen. The spongosa, which is the middle layer, is going to be mostly composed of gags. And then your atrialis is mostly composed of elastin. And there's 2 main cell types on these valves, and those are the endothelial cells, which are kind of throughout the leaflet, and the interstitial cells, which are more centered in the leaflet. And so we focus on children with congenital heart valve defects, and these are children that have problems with the structure or function of the heart that's present at birth or soon after birth. And this causes abnormalities in the blood flow, which will lead to regurgitation or stenosis. And so you can see here, regurgitation is when the valve does not close properly. So you're gonna have this backflow that's always occurring. And stenosis is when the valve does not open properly. So you're not gonna have enough blood that's gonna be flowing through. And again, we specifically focus on children with critical congenital heart valve defects, specifically in the mitral valve. And these are patients that need a proper treatment within a year of their birth. And so the treatment options for these patient population is lacking. And this is because currently what's available are mechanical or bioprosthetic valves. And these do not come in small enough diameters for these patients, as well as they do not allow for somatic growth. So they don't grow with the patient over time, which would lead to multiple reoperations. And so again, there's no treatment except for experimental or compassionate care option for these patients. And so this is why we think a tissue engineered heart valve would be an ideal treatment for these babies, because they will be able to grow with the patient over time. And so we're specifically interested in porcine small intestinal submucosa, which I'll be referring to as PSIS. So this is just a bioscaffold that comes from the pig's small intestine. And it's a decellularized bioscaffold, meaning that all the cells are removed from the scaffold. And so you're basically left with an intact bioscaffold that has the ECM or the extracellular matrix components. And this is FDA investigational device exemption approved. And what's interesting or nice about this bioscaffold is that it already has the structural components that are needed for valves. And so we were interested to determine whether this PSIS would be a viable option for these children. And we wanted to see that it functions robustly, that it would allow for somatic growth, and that it would remain biocompatible. And so in order to do this, we looked at raw PSIS, which is just the bioscaffold without any treatment, and implanted them in 2 baboons, and then would assess them over time. And then we also looked at the PSIS seeded with stem cells and exposed to flow in our bioreactor system to see if we can produce some sort of ECM and then implant those into baboons as well. So I'll talk to you guys about some of our results from these. So as I mentioned, we started off by looking at just the raw PSIS, not treated. And what we would do is we would take echoes of our baboons right before surgery to get the dimensions of the valve that we needed. And so we would make them by hand. And here is a quick video demonstrating how it works. So we would get the dimensions and then mark the length we needed and kind of suture here along until we get to the length we need. So then once we get there, we tie a bunch of little knots so that the valve doesn't become loose. And then we get one side and kind of flip it over to make it a thicker area to make that the annulus of the valve. And so then when we're done, we get the length again that we need and then cut out the excess amount of the valve and then cut off the sutures. And then again, we always remeasure, make sure we have the right dimensions, and then cut out the extra length as well. So this that you're seeing here is the bioscaffold that we would be implanting into the baboons. And so once the valves are made, we place them into the mitral position of juvenile baboons or baby baboons. And we suture one of the legs onto the papillary muscle, as you can see here, and the other one into the side of the wall of the heart, and then around the annulus. And then this is a very quick video showing already, you can see here that the valve has been implanted, and they're kind of suturing around the annulus here now. And so once we implant these, we do monthly echoes on the baboons. And so with this, we can look at the in vivo growth potential. And we did this by calculating this novel parameter that is the normalized aspect growth ratio, or the NAGR. And this is essentially just looking at the growth of the valve over time compared to the baboon. And so an ideal growth would be equal to 1, where they're kind of growing at the same rate. And so we looked at our baboons, and over time, you can see that in the beginning, our NAGR was ideal, where it was equal to 1. But over time, we do see this decrease. And we think it's due to the fact that the baboons are growing at a quicker rate than the valves. And so with these monthly echoes, we also look at the functionality of the valves. And so you can see that over time, the valves function well with trivial to mild regurgitation. And so then we looked at our explants of our valves, and we were looking at the de novo tissue of the ECM, as well as the cellular repopulation. And so just looking at these raw images, again, this is the valve that we implant. And then you can see at our 3-month, 11-month, and our 20-month explants that we are already starting to see a tissue formation. And also we're starting to see this nice integration in the papillary muscles, as well as at the annulus area here. And so we wanted to do histological stains so we can see the ECM content. And so here we're doing a MOVOT stain, and we're looking at the native baboon valve, the 3-month explant, the 11th-month explant, and the 20-month explant. And with MOVOTs, you can see kind of this greenish yellow color is going to be your collagen. The blue color is going to be your GAGs, and then this purple, almost black color is your elastin. And so you can see that compared to the native baboon, we're starting to see all these ECM proteins at all of our explants. And then we wanted to be able to quantify the amount of ECM proteins. So we did spatial intensity maps to be able to quantify them. And so this is an example of our spatial intensity maps, where we're looking at collagen, elastin, and proteoglycans. And here this is of the native baboon valve. And so we got the average percent ECM components, and this is looking at the leaflet location, again, at our native and then our explants. And we found that there was no significant difference between these ECM proteins at any of these explants. And then we kind of did the same process, looking at the percent ECM components, but this time looking at different areas of the valve. So looking at the annulus area and then the neocord papillary muscle integration area. And so for the annulus, we saw that there was no significant difference here as well, except for at our proteoglycans at 3 months, we saw that there was a significantly lower amount of proteoglycans. But then at our neocordiate papillary muscle integration area, again, we see that there's no significant difference here. So overall, our PCIS valves developed the ECM components that we were seeing in the native baboon. But then looking at another stain, which is H&E, this will give you the morphology of your tissue as well as the cells that are there. We did H&E on, again, our raw PCIS, so the one before implantation. And you can see that there are cells remaining here that you see the purple little dots everywhere. And again, this is coming from the pig's small intestine. So the cells that are remaining here are pig cells. And so we looked at the native baboon here, the leaflet, And you can see that there's this nice morphology. You see the normal cells here. And then looking at our explants, at our 3-month here, we see that there's this morphology that's kind of forming, which is nice. And we do see that there's some cells that are infiltrating here onto our scaffold. And then for our 11th month and our 20-month explants, we did see areas that did have this nice infiltration and tissue formation, but we also saw that there was a chronic immune response. So we think it's because of these darker pink areas here, which again are these residual pieces that have these porcine cells still on the scaffold. So we're seeing this chronic immune response anywhere that there's residual pieces. And so we got the average percent of the residual pieces on our scaffolds. Again, looking at different areas of the valve, so at the leaflet, the annulus, and the neocordiate areas. And you can see that just with these small percentages that we are having this chronic immune response occurring. And so this is why we believe that seeding our pieces bioscaffolds with stem cells and then conditioning them in our bioreactor with flow will be able to eliminate the residual pieces that is causing this immune response. And then it would enhance the tissue formation that we would want. And so in order to do this, we developed a new torpedo bioreactor in our lab, and it accommodates the tubular valves that we'll be using. So here you can see our bioreactor. So this would be the inlet, and this is the outlet here. And you can see the flow would be going this way, and this black kind of rod torpedo that you see is our torpedo area, where we put our scaffold on top, and then these green little dots represent the stem cells. And so this is an image of two of our valves that have been seeded and placed in our bioreactor. And you can see that we were able to have our stem cells seed on our scaffold at a high density, where you can see all the purple little dots everywhere, which are the cells. And then we also saw that our stem cells were able to differentiate into both valvular cell types. So here, endothelial cells, which you can see kind of in this greenish color throughout. And then our interstitial cells, which are kind of this pinkish, almost orangey color. And then we also did a MOVOT stain here to see the ECM proteins that we could produce. And again, here we were looking at the spatial intensity maps to be able to quantify the ECM content. And so we found that our dynamic group, which is the group that we seeded our scaffolds and placed them in our bioreactor, was able to produce a significantly higher amount of all of our ECM proteins, except for collagen, where collagen was pretty much the same at both our static and our dynamic groups. And here our static group is just the pieces that is seeded with stem cells and then left in static conditions. And again, our dynamic is the bioreactor group. And so we also calculated the percent of de novo tissue, and we found that we were able to get a significantly higher amount of tissue production in our dynamic group. And so what's important here is that elastin is not easily produced in vitro at all. And we were able to produce elastin in both our static and our dynamic groups. And again, our dynamic group, we were able to get twice the amount. And so given all of these findings, we believe that our tubular pieces valves, when they're seeded with these bone marrow stem cells and exposed to flow in our bioreactor system, we would be able to create this coat on our valves of a thin layer of robust tissue. So that way we'll have less residual areas of pieces when we implant, and then we can mitigate the chronic inflammation and enhance tissue formation. And again, our end goal here is to use this as a permanent approach for these children with critical congenital heart valve defects. And so currently, we are in the process of creating 2 more of these valves, but this time for implantation. So these are growing in our lab now as we speak. And we are going to be implanting them in 2 weeks to 2 different baby baboons. And then we will be assessing them over time to see if these enriched ECM valves will actually mitigate the chronic response that we're seeing, and then help grow tissue at a quicker rate. And then as well, we'll be looking at the functionality of these valves over time. And so with that, I'd like to thank my advisor, as well as the CV Putics Laboratory, specifically these people in our lab that helped with the project, as well as the surgeons and cardiologists at Joe DiMaggio's Children's Hospital, and then the vets at Core Veterinary and Mannheimer Foundation that kind of house the baboons and keep them on constant monitoring, and then our funding sources. And so with that, I will change the floor to Hooi.
Can everyone see my slides all right?
Yes.
That's great. So first, I would like to thank the BME department for the invitation to share our work on targeting the relaxin receptor as a novel therapeutic strategy for vascular calcification. So as we know, the deposition of calcium mineral in cardiovascular tissue is now accepted as the most significant predictor of acute cardiovascular events. Vascular calcification is often encountered in advanced atherosclerotic lesions, and it has been reported that in chronic kidney disease patients, they exhibit 2 to 5 times more calcification than the healthy age-matched individuals. And currently, there are still no effective pharmacotherapy for the treatment of vascular calcification. So my research has centered around the peptide hormone relaxin for the past 8 years. So what is relaxin? Traditionally, relaxin is known as a reproductive hormone that mediates cardiovascular and renal adaptations to pregnancy. And over the past few decades, relaxin has been shown to exert vasodilatory effects, anti-fibrotic, anti-inflammatory, as well as cardioprotective properties in various organs. Relaxin binds to and mediates its action through a G-protein coupled receptor, RCEP1. So here we are looking at the localization of the relaxin receptor, RCEP1, in red vessels on the left. So this is just a semi-quantification of the distribution of RCEP1 in both the endothelial cells as well as the smooth muscle cells. So in red aorta, we can see that RCEP1 is more abundantly present in the endothelium in comparison to the smooth muscle cells. Whereas in the femoral artery and vein, RCEP1 is more abundantly present in the smooth muscle cells in comparison to the endothelium. But in the mesenteric artery and vein, as well as the vena cava, RCEP1 is more abundantly present again in the endothelium in comparison to the smooth muscle cells. So I then follow up with this study and showed that RCEP1 is expressed in both the endothelial cells as well as the vascular smooth muscle cells of the male's aorta, as shown in the brown staining in this immunohistochemistry image. So in recent years, we have shown that serelaxin, which is the recombinant human relaxin 2 treatment, was able to reverse vascular dysfunction in type 1 diabetic mouse, as well as reduce endothelium-derived vasoconstricting factors in the spontaneously hypertensive red mesenteric artery, as well as the red aorta. Additionally, we also showed that relaxin treatment was able to reduce oxidative stress to attenuate nitric tolerance in the mouse and red aorta. And importantly, relaxin has been used in clinical trials for acute heart failure treatment. So in this trial, intravenous infusion of relaxin in this patient was able to reduce the symptoms dyspnea, which is like shortness of breath, and it was associated with a reduction in all-cause mortality in this acute heart failure patient. And most importantly, intravenous infusion of relaxin in humans was shown to be safe. However, relaxin poses the universal drawbacks of peptide-based therapy, such as poor oral bioavailability and short in vivo half-life of approximately 10 minutes. So to overcome these challenges, we have generated a non-peptide small molecule agonist that selectively targets the RZP1 receptor, and this molecule is named ML290. Unlike relaxin, which can activate various mammalian species RZP1 receptor, ML290 can only activate the human, monkey, and porcine RZP1 receptor. Therefore, we have to generate a new transgenic line of mice which express the human RZP1 receptor. So with this transgenic line of mice, we were able to test the efficacy and potency of ML290 in vivo. And more recently, we have shown that ML290 has excellent oral bioavailability and it has longer in vivo half-life in comparison to relaxin of reported 8 hours in the plasma of mice. So based on the previously published findings on the vascular protective actions of relaxin and these excellent properties profiles of ML290, we therefore aim to test the hypothesis that ML290 treatment can arrest the progression of vascular calcification in mouse models of atherosclerosis and chronic kidney disease. So in the first study, we crossbred our humanized mice which express the human RZP1 receptor with the ApoE knockout mice. So these ApoE knockout mice, they had deficiency in cholesterol metabolism, which made them prone to develop atherosclerosis. And these mice were then put on an atherogenic diet for a period of 25 weeks, and during which they received either vehicle or ML290 at a dosage of 30 milligram per kg via oral gavage every other day. And we also have a negative control group, which is a group that receive normal lab chow, and these animals, they receive vehicle via oral gavage as well. So at endpoint, at 25 weeks, we injected a near-infrared dye into the tail vein of this animal to look at the total calcification burden in the aorta of these mice. So I'll just briefly walk through how we get these vessels. So at endpoint, we kill the animals and we take out the heart as well as the aorta. So the heart was initially attached to the aorta, and we clean off the adventitia as well as the perivascular adipose tissue to get these images. And this graph is just showing the quantification of percent positive signals of these osteosense dye, and we normalize the data to the total aorta area. And as you can see here, the brighter signals indicate there's more calcification and dimmer signals indicates there's less calcification or no calcification. So we can see here on the quantification graph, there's a significant increase in calcification in the aorta of these vehicle-treated atherogenic mice. And our treatment with ML290 every other day over a period of 25 weeks was able to significantly reduce this calcification in the mouse aorta. So we then move on to look at the temporal changes of mineral formation in a single mouse with the same schematic. So in this study, we injected different fluorescent dyes that have distinct properties at different time points over the period of 25 weeks. So in blue here, we can see that we injected calcium blue via intravenous injection. And at 15 weeks, we injected calcium green. At 20 weeks following atherogenic diet, we injected alizarin red. And again, similar as the previous scheme, we injected osteosense at endpoint. So here on the image, we are looking at a 3D reconstructed confocal image that shows calcium green, alizarin red, as well as osteosense signals in gray in the mouse aorta. So you'll realize here that we did not observe any calcium blue signals, and this suggests that there was no calcium accumulation during the first 10 weeks on atherogenic diet. And here on the right image, we can see that the signals of calcium green, alizarin red, as well as osteosense was significantly reduced by ML290 treatment. So collectively, this data suggests that our treatment ML290 slows down the progression of calcification in the mouse aorta. So we then move on to ask the question whether can ML290 reverse atherosclerotic calcification. So in this study, we start our ML290 all vehicle treatment following 15 weeks on the atherogenic diet. And again, we used the similar dosage as previously mentioned, and these mice were given ML290 or vehicle every other day via oral gavage. And similarly, at 25 weeks, we injected the near-infrared dye into the tail vein of these mice to look at the total calcification burden in the aorta. And here we can see that ML290 treatment start at 15 weeks. It was able to again significantly reduce the aorta osteosense signals, suggesting the reversal effect of ML290 in these atherosclerotic mice. So we adopted similar approach as previously mentioned to look at the temporal changes of mineral formation in a single mouse. And here on the right with ML290 treatment, we would expect to see similar calcium green signals in this ML290 treatment because we didn't give any ML290 or vehicle to these mice before 15 weeks. But interestingly, we did not observe any in the aorta of these mice. So this suggests that ML290 may potentially promote mineral resorption in the aorta of these atherosclerotic mice. So now we know that atherosclerosis-induced vascular calcification is a very complex process, and it involves various inflammatory cytokines as well as macrophages infiltration. And we really want to tease out whether it's ML290 selectively targeting the smooth muscle cells or the medial layer to prevent or reverse vascular calcification. So in the next study, we adopted a fairly new mouse model that was published in 2017. And this model, they use chronic kidney disease as the disease model to induce medial arterial calcification. So again, we use the humanized mice that express the human RSIP1 receptor, and these mice were put on a special diet that contained 0.2% adenine for the first 6 weeks, and then these mice were then changed to a high phosphate diet. Again, they are still on the 0.2% adenine to drive the medial arterial calcification process. So here we can see that ML290 or vehicle were treated following 6 weeks of chronic kidney disease, and they were given daily on this drug. And again, similarly, we use osteosense to look at our total calcification burden in the aorta of these mice. So on the left here, we've got vehicle-treated child animal, which is the normal diet of our mice. And in the middle image here, we are looking at the mice that receive the CKD diet. And on the right here, the mice that receive the CKD diet and treated with ML290 daily at 30 milligram per kg via oral gavage. And we can see that those mice that were put on the high phosphate diet, they've got increase in aorta osteosense signal, suggests medial arterial calcification. And ML290 was able to prevent this phenotype. So we then move on to look at the in vitro effect of ML290. So here we are looking at the primary human coronary artery smooth muscle cells, and these cells were treated with osteogenic media. So what is osteogenic media? It is a media that's typically used to induce bone-like phenotype in vascular smooth muscle cells. And we can see here in the vehicle-treated vascular smooth muscle cells, we can see a lot of red staining, which is the alizarin red. So this suggests that there was an abundant accumulation of in these vehicle-treated vascular smooth muscle cells. And with ML290 treatment at 5 micromolar, the effect was significantly abrogated, suggesting the anti-calcification effect of ML290 in vitro. And this effect was associated with a reduction in superoxide level in these primary human coronary artery smooth muscle cells. So if you recall from my introduction, I mentioned that ML290 can activate human porcine as well as monkey RSF-P1 receptor. So we then went on to look at the effect of ML290 in the porcine coronary artery smooth muscle cells by looking at the mRNA level as well as the alkaline phosphatase activity in these porcine cells. So here we can see ML290 treatment at both 5 and 1 micromolar. It was able to significantly reduce or prevent the increase in several osteogenic markers, including Relax2, NOX4, BMP2. And here on the right, we can see a significant reduction with ML290 in the alkaline phosphatase activity in these porcine SMCs. So my mentor, Josh, he has previously published that these small extracellular vesicles excreted by these smooth muscle cells are very important in mediating calcification. And we are interested to see whether is there any effect of ML290 to prevent the formation of these calcifying extracellular vesicles in vitro. And again, we used our model of primary human coronary artery smooth muscle cells and look at the calcifying EVs that are released by these human cells. And in fact, ML290 at both 5 and 1 micromolar was able to significantly reduce the calcifying EV formation in these culture. So in summary, we have shown that ML290, a small molecule and is a non-peptide agonist at the human RSF-P1 receptor, is able to prevent and reverse vascular calcification. And more importantly, ML290 poses excellent ADME profiles, and it has low cytotoxicity. And we have shown that it is safe and well tolerated in our atherosclerosis and chronic kidney disease mouse model. And with that, I would like to thank my mentor, Josh, as well as our entire team in the cardiovascular matrix remodeling lab for their intellectual input, as well as technical assistance during my studies. And we also like to acknowledge our collaborators for kindly providing us with the drug ML290 for our studies. And last, I would like to thank our funding sources, including the American Heart Association, FIU Foundation, Florida Heart, as well as the NIH for supporting our work. And thank you for listening, and I'm happy to answer your questions.
So I ask people to pose questions in the Q&A. At this time, I don't see any questions that you might have. That would be good to get from the community that is listening. Maybe I will start with Brittany, that you're talking about, when you are, I was going to ask you, but I think somebody put a question in chat, put it in Q&A so that we can follow that. And Sweta Gupta asked, can magnetic field help in decalcification?
I'm not sure about that. We haven't looked at any magnetic field, so I'm not sure about that.
Okay. And Dr. An anonymous attendee asked to Brittany, I was wondering how you overcame that fear of dissection and blood and stuff.
That's a great story. So I was told that we were going to a slaughterhouse to get the heart and dissect the valves, but I was told I was going to be watching. So I had mentally kind of prepared and I was ready to just watch and kind of squint a little bit, not really watch. And I got there and the person told me, okay, you're dissecting. So I kind of just was thrown in there and didn't have an option. So my first dissection was not the greatest because I was kind of like not looking fully the whole time, trying to not smell the nice smell that was coming in. But after time, I just got used to it, and now watching the surgeries of the baboons and doing the dissections, so I just kind of got used to it.
Great. Dr. Hutchison asked both of you, very nice work by both, and I will second that. I think all of us can second that. Kudos to that. And he asked both of you to comment on how we might move the research from the lab to clinical utility, both for Hooi and Brittany. And as you are answering that, I would also, I would like to add on to this thing as you're talking about it. Both of you are talking to some extent of what is happening in valves. And so I would like you to reflect on the other person's work and see how you utilize that kind of work to link yours. So how might we move the research from the lab to the clinical utility? And how could you take information from another person's work in that area and link with yours?
Do you want to start first, Brittany, or do you want to start first?
So I think to move things from lab to clinic, it's really important to have collaborators, either with industry or with hospitals, or collaborators outside of just research, because that's what helps everything kind of move towards the clinic, as well as, of course, different grant agencies. But I think at least in my work that we're working on, since we have our collaborators at Joe DiMaggio's Children's Hospital, as well as the funding that we need, and also collaborators that are vets and everything, we do have a lot of push towards this going kind of clinical in a couple of years, maybe 5 years-ish, if we can get all the data that we need. And I think that's mostly because, again, of our work here in the lab, but mostly because of our collaborators at Joe DiMaggio's Children's Hospital.
Well, I guess for me, it'll be quite different because we are testing a small molecule that has never been used before, and we don't know the safety profiles in humans at this point. That's why I keep mentioning about the safety profile of ML290, at least in our mouse model, which is like a preclinical phase. So there's still a lot to learn about this small molecule before moving it into the translational phase of research. So I'll say the first step is to make sure that the drug or the compound that we are testing is safe and tolerable at least in preclinical model at this point, before moving into the next phase.
And I can also add that, at least for our work, we actually have the collaborator of the industry working with the doctors as well. So they've actually had these valves placed into pediatric patients to see how they work in them, because again, it's FDA approved for early assessments. So we're able to kind of see how it's working in our baboon model and kind of enhance it, but the earlier version has been implanted in some pediatric patients and kind of following them to see how it goes.
Dr. Ramaswamy asks, Hooi, nice work. can this therapeutic have potential for valve calcification? Any preliminary data on this? And he says this is because vascular versus valvular calcification have different potential mechanisms.
So I did actually look at the valve of both models. So I'll just start with the ApoE knockout mice. I did not observe a lot of calcification, if any, in the ApoE knockout animal models on high-fat diet. But on the other hand, with the CKD model, we observe quite a significant amount of calcification in the aortic valve, as well as the mitral valve of those CKD mice put on high phosphate diet. And with preliminary data with ML290 on the valve aspect, I would say yes. I haven't finished the analysis yet. So the pilot data suggests that there was a kind of a reduction with calcification in the aortic valve of those CKD mice.
Okay. So I have a question for Brittany, and different valves, like you have a particular valve with certain flow chambers and all. Can you comment upon different valves and different aspects about those of what these flow profiles may do? And are there other disease models, congenital disease models in particular, that one may be interested in looking up, and how your knowledge may transfer to that?
Yeah, so there's the 4 different valves, but usually we look at the left side of the heart because those are the ones that have the higher flow and pressure. And those are the ones that are usually affected the most with disease. So there's going to be a higher pressure in those valves. So that's why we focus on the left side, specifically the mitral valve. We focus on just because we have patients, like the children patients, that had those implanted in those areas. So we wanted to kind of compare with the implants that we had.
And then for Hooi and for either of you, you are looking at certain mechanisms for the vascular calcification in a particular area, which is in the heart. Can you say something about other blood vessels in the body and impact on other disease states?
Maybe I can start with this. So yes, we look at the effect of relaxin in various vascular beds, and the effect is quite different, I would say. So with relaxin treatment, sometimes we see significant reduction in vasoconstricting factors in the aorta, and sometimes it's in the messenger arteries. So it's like the large vessels versus the resistance vessels. But we haven't get the chance to look at these with ML290 treatment yet. So
what about things in stroke, middle cerebral artery, or things like that?
Yes, with relaxin, but again, not with ML290. I think this is the first study, to the best of my knowledge, this is the first study that look at ML290 effect in the vascular system.
All right. Can either of you comment further on, and maybe Hooi can comment a bit more about what you might see as a role for multi-scale modeling, computational modeling, as a role in your work? And also, Brittany, obviously you're doing a little more on that end, but if you could comment upon the role of before doing things in your actual how am I blanking on the name, where you put all your stuff in, the
bioreactor.
The actual experiment, rather than the actual experiment, doing a model version of that.
Yeah, I think it's a good approach, and I have no experience in computational modeling, to be honest, but I think it's a good way to start looking at the efficacy or the effects of drug in various blood vessels or various organs.
Yeah. For me, I haven't done computational work either, but I know specifically for our valves, we do some CFD or computational fluid dynamics to get the proper flow that we would need for our bioreactor. So I know that in my lab mate does the computational fluid dynamics assessment before I actually make the valves. So we've kind of incorporated that in our system.
Do either of you see then in your next steps in your future making a partnership like that for when you are applying for grants, for example, where you might partner with a computational or theoretician, engineer, another biomedical engineer, as part of your team that is besides the clinical folks, as part of as a key member of your team to move your research forward?
Yeah, I'll say absolutely. So in my first study with the ApoE knockout mice, we actually used about 6 to 7 months to generate the data. So I think during that period, it'll be good to team up with folks from modeling area to look at the effects of the compound or the drugs that you're interested in.
Mm-hmm. Yeah, I mean, I second that. I think it's great to always have collaborators that are in different expertise than you and kind of collaborate together to get one goal out.
There are 2 more questions. One question is to you, Brittany, from Dr. Hutchison. In tissue engineered constructs, how close do you think we need to be to the native valve before implantation? What are the important properties that must be met?
Good question. So I think we're getting closer and closer, but I still think there's a lot of work to be done in the field itself. I would say we would have to have kind of, I guess, the same morphology based on ECM, I guess, having the right components, as well as once implanting, seeing that like cells actually go to that area. So I think there's still a lot to do, and it's kind of hard to pinpoint what exactly we would need, because in one patient it can be totally different from another patient. So I think we're getting closer, but I still think there's a lot of work to be done.
And then both of you, if you could please comment to this anonymous attendee's question. Can you comment on how it feels to be a female researcher in a male-dominated field? And by the way, there is yesterday and today, there is an ongoing national meeting going on, which you might be aware about, of equity and anti-racism transformation in BME. So we had a fantastic session all day yesterday. And then when I get off of here, I'm actually as a panelist in one of them. So I want to hear from both. I also want to hear this, but I think obviously somebody else wants to hear about it.
Do you want to start, Hooi?
I guess just don't be afraid of anything and go for it, right? Or at least that's my attitude of like doing science in this field.
So I think the question, a lot of the question is, how do you feel? It is about your reflection of how do you feel as a female researcher in a male-dominated field?
I guess I'll go. I never really, I guess, thought anything of being female or male, because I've always been so focused on like the research, and being in like my lab or Dr. Ramaswamy's lab, I'm kind of surrounded equally amount by male and female. I would say we actually have more females. So I never really felt so much of the differences in sex, at least here. But I do know in my undergrad, I did feel it a little bit more, because there was mostly guys around me. And I think that never really stopped me, but it always made me work a little bit harder, because I know being in like groups where I was the only female, I would be speaking, but sometimes they wouldn't listen, not listen, but they would just kind of, they would listen to what I was saying, but like not really give an opinion about it. So then like later on, if someone would finally come to the same conclusion as me and say basically the same thing, they'd be like, oh great idea. And I would just sit there being like, oh, awesome. I said that 2 weeks ago. So it was kind of just me trying to voice my opinion a little bit more, and trying to work a little bit harder, I guess. But that I saw more in my undergrad. I think once, at least for me, once I came into my professional degree, I think that you're a little bit more well respected, I guess. So I didn't see that as much here. But that's just my experience.
Yeah, the same goes for me. From my previous lab, there's always more females than males, and I don't encounter any issues with that from my personal experience, at least.
I'm very glad to hear that, that you have had your current experiences, at least in our BME department, have been very strong and positive, and it is kudos to the role model as that your own male mentors and advisors have been, and hopefully the climate around is good. But as we all know, the condescension, the exclusion, if kept on, can be deafening. And if I may use Dr. Martin Luther King's words in a different context, he said, "In the end, we will remember not the words of our enemies, but the silence of our friends." And so I hope that others who might be in a scenario like Brittany that you saw would break their code of silence and not let that happen, and be proactive in making sure that everybody has their that there is this opportunity to voice what you do, and give due recognition early on. So that's fantastic. With this, we had you both of you have done a fantastic launch of our of this series for us, that early career investigator series, and we are hoping that we will bring other members of our own BME community in here, and let us see where it goes, and whether we might invite outside members also to come in as early career investigators to speak at this series. So any input you guys can give us and feedback on this new launch of the series would be much appreciated, that we can go forward with. Thank you for educating us. Thank you for doing the what is it called, the band that plays before the main act of the getting us launched to the Miami Heart Month. That is that is what you are doing today for the next whole month. And so now I hope you will have an opportunity to talk to the students, grad and undergrads at noon. Okay, thank you.
Bye. Thank you.
Thank you for having me.